It is generally believed that protein search is a downstream task, or a special case, of representation learning. Given a query, rank the protein database by biological relevance. So why is this worth paying attention to? First, from an application perspective, protein search can retrieve proteins with low sequence similarity, but highly similar functions. Second, this allows other protein engineering methods to escape local optima and provides them with better starting proteins. The technical roadmap looks like this. Step 1: Traditional retrieval. Rely on fixed rule-based scoring. Seeking a balance between speed and hit rate. Representative tools include sequence 2, fold-seek, etc. Step 2: Enhanced by PLM. First, use a protein language model to learn sequence information, then transform it into a similarity network to predict the similarity between query and target. Representative tools include PLM search, PLM align, TM, VEC, etc. Step 3: Sequence structure. Bilingual models. Discretize structure into something like structure tokens, enabling bidirectional translation between amino acids and 3D. Representative approaches include PROST, T5, Fold Explorer, etc. Step 4: Multimodal and larger external knowledge. Align sequence structure text into the same system. Further down the road, a more engineering direction. Treat massive databases as external memory. Let Elm use tools to continuously retrieve, decompose tasks, and aggregate evidence. In the past, finding proteins was like searching by name spelling. If the spelling didn't match, you couldn't find it. Now with AI, we can find distant relatives through function and structure, even if the name is completely different. So what do we need to consider when modeling this? First, data scaling. Use prediction models or new benchmarks to construct label, reward datasets. Also leverage folding, inverse folding, and bio-llm.to fill in cross-modal data. But pay attention to the necessity of multi-dimensional filtering. Second, modality fusion. Compare the pros and cons of the two current mainstream modality fusion approaches. LLM-based multimodal naturally has strong instruction following ability, but suffers from catastrophic forgetting after synthetic data training. Precise hyperparameter control is needed. Symmetric multimodal modeling unifies the space so any modality can be translated to any other naturally. No need for auxiliary prompts. But due to data distribution differences across modalities, alignment is very difficult, and inconsistency is common for modality fusion algorithms. Local features like binding sites and functional domains are more suitable for cross-attention blocks. Global features like family, evolution, and cellular distribution are more suitable for contrastive learning or clustering. Finally, the importance of validation. Protein search is particularly AI-friendly because in many cases, it can be evaluated offline. However, if you want to aim for top-tier publications, many scenarios still require wet lab experiments to prove the things we found are truly useful. This often determines the difference in paper impact. We can compare representative learning and de novo design to intuitively understand the goal of protein search. The core objective of protein search is to learn a biologically meaningful similarity and distance function DQP that judges the evolutionary relatedness between two proteins. It differs from de novo design, which learns a generative function to create new proteins from random noise that fit the natural distribution. It also differs from representation learning, which learns an encoding function that turns proteins into vectors preserving biological properties. Protein search is more like searching for proteins with an evolutionary perspective, compared with web search. First, from the input perspective, protein search modalities are naturally correlated. The model needs to automatically complete missing modalities from a single modal input and jointly reason about relatedness. Second, input is a concrete functional or structural requirement. Output is high confidence evolutionary relatedness hypotheses. Third, matching is based on true biological evolutionary kinship, not textual semantic similarity. Fourth, the database has missing modalities, extremely uneven distribution, and many outliers, while web databases are broad, but noisy and loosely structured. That basically wraps up the summary. If you want to dive deeper into the details, feel free to join us for a more thorough analysis and discussion. We will share our parsing and discussion process with you into parts.